Excellent. And then, yeah, I'll probably just start with introducing JETZON and myself. So this is the JETZON call for February, although it's the last day for me at least. JETZON is a program that brings together, or an initiative, but it brings together different programs that are related to mesopelagic research, for example, carbon flux or biodiversity, or different research regarding the mesopelagic, because we think it's a really understudied part of the ocean. So yeah, welcome. And every month we organize these JETZON calls, and usually at the time, so there's a time slot in the Pacific, this one, and then the other one is organized by Adrian Martin in the UK, who is organizing a time slot more appropriate for the European and I think East Coast US audience who don't want to get up in the middle of the night. So we kind of swap in between these time slots, and the good thing about it is if you miss a call, you can always look them up on the JETZON YouTube channel. And maybe before we go into the talks, and this is I think the main thing, and then I stop talking, is that I want to mention that JETZON is organizing a workshop in mid-September. It's WHOI, probably in collaboration with the there's an Ocean Twilight Zone program workshop also in the middle of the September, and afterwards there's the JETZON workshop, and hopefully we can, after years and years of online talks and meetings, which is great, but finally get back into a room all together and meet everybody. So yeah, keep your eyes peeled. There will be a last information confirmed. That our last information is confirmed just now. So yeah, there will be some announcements, I guess, in the coming months. And also there will be travel funding for ECRs. There's some grants you could apply for. So I think it's I mean, in my case, I haven't been to the US before, so I'm really excited to actually go and present some stuff and meet all the people, network again finally in person. And another thing is, if you're interested in fisheries and carbon, there's a workshop you can probably still sign up next week. It's on the OCB website. I can have a look. It's fish fisheries and carbon, all about fisheries and carbon interactions. So I put the link in the chat, but that's just an interesting thing going on next week. And then yeah, I think let's start with the actually interesting part of the thing here, the call. Maybe Alia, just let you introduce yourself. I think it's the easiest.
Sounds good. Thanks, Venya. Hello, everyone. My name is Della Penna. I go by Ali, and I'm a lecturer at the University of Auckland. And I'm really pleased to be here and present some of the results from my recent work. Before I start, I'd like to acknowledge my co-authors. As I was mentioning earlier, we all started when we were at the University of Tasmania, and most of us went on a voyage together, and then the collaborations kept us connected, even if now we're sort of all spread around the world. So today I will be presenting a study where we looked at how Southern Ocean eddies might affect mesopelagic fish. So let's see if this moves forward. Yeah, so I'll be referring to mesoscale eddies. And with this term, I'm referring to rotating currents that typically have a spatial size from tens to hundreds of kilometers, and that can persist from a few weeks to a few months. These are the oceanic equivalent of weather patterns. As we have cyclones in the atmosphere, we have the same structures in the ocean. They're just smaller and slower because water is denser. And we can often see them in a near-surface chlorophyll distribution. This is an example of a ocean color image shot sort of in the Tasman Sea between Tasmania and New Zealand. And we can see some rotating structures that have been trapping and moving around phytoplankton. But these eddies actually have a signature that extends through the water column, going more often than not as deep as 1,000 meters or even further, so right into the mesopelagic. And while we're learning more and more about how these structures can impact the near surface and phytoplankton, we're also accumulating observations that suggest that apex predators such as marine mammals, sharks, billfishes, and some seabirds tend to spend a lot of time around eddies, and they alter their swimming and foraging behavior when they're in the region. And one of the reasons why we think that happens is that because the eddies are somehow structuring the prey of these animals. A lot of the prey of these apex predators are mesopelagic animals, especially micronecton. So the question is, how do eddies affect these organisms? And there isn't a lot that is now known about them. So I'll be referring to this very broad label of small animals that are a few centimeters or a few inches in size. Here there are some examples of small fish, squids, crustacea. There is some zooplankton in the image just because I think they're beautiful and they should be in every talk. But they're extremely abundant. So there is a huge biomass of this. They can swim. They're not plankton, so they have some movement. And as mentioned earlier, they are very important for prey items for large animals, but they also play a key role in the ocean carbon cycle because many of them migrate into the water column. So they're moving carbon up and down into the deep ocean. So even if they're very important, we don't know much about these animals because they're really challenging to study. And in particular, where we're learning more and more about their distribution at a basin scale, there is very little that is known about how eddies and mesoscale circulation affect the distribution of these animals. And this is where I think this study can make a bit of a contribution. I'll be presenting some results from a voyage that was called the Eddy Voyage that took place in autumn 2016. So it was April because we're in the Southern Hemisphere. We left from Hobart, Tasmania, and we crossed the subantarctic zone and got into an area close to the subantarctic front, that is this black line here, where a cyclonic eddy, that is marked here as this blue little patch, had detached from the front about a month before of our sampling. So the water inside the eddy was originally from the subantarctic front and the polar front zone on the other side of the front. So this eddy was tracked using satellites. Here there's an example of sea level anomalies. And the main goal of the study was actually to understand the impact of the eddy on the biogeochemistry and transport of physical properties such as heat and salt. So it wasn't really about fish, but the sampling was really strategic because we sampled the eddy core. So here we have a zoom in of the area where we conducted our sampling. The black line is a SLA isoline just to keep track of where the eddy was. So we sampled the eddy core here in red, but also the periphery here in yellow in a star-shaped pattern. We also sampled quite extensively the subantarctic water zone in blue here. And crucially, the area where the eddy had originated 1 month before our sampling here near the polar front zone. And really luckily for me, then we had a really good characterization of the ocean physics of the eddy. Here I'm just showing a section that was put together using the CTDs that were carried out regularly. And you can see here the distribution of temperature across different depths. And you can see that the doming of isopycnals, that is a typical signature of cyclonic eddies, extended all the way to 1400 meters. So we're really deep in the mesopelagic. Luckily for me, for the entire duration of the trip, a scientific echo sounder pinging at 18 kilohertz was on. So we had observations from active acoustics through the entire sampling. So for those who haven't used this kind of observations before, the idea is that you're sending sound down the water column. And when sound encounters something that has a different density, is reflected back. And by measuring the intensity of the reflected signal and the time difference, you can get a sense of where things are in the water column. Things could be the seafloor, they could be the thermocline, but also the swim bladder of a fish or the shell of a small zooplankton. So this is a tool that is often used to look at the distribution of zooplankton, but also micronecton, especially at this lower frequency in the water column. what we found when we looked at the data were basically a set of echograms. So this is an example of one where you have time on the x-axis, depth on the y-axis, and the different colors here indicate the intensity of the reflected signal. And generally, the reflected sound is organized around deep scattering layers. I don't know if you can see my mouse, but here I'm around 200 meters, thanks. That can be at different depths in the water column. And there's also a typical behavior that we can see where there is a diel vertical migration where animals tend to spend more time near the surface at night, so between sunset and sunrise, whereas during the daytime, there isn't much activity near the surface and animals tend to be present deeper in the water column. So this is a very common pattern that we see in echograms, but it's not the one I'd like to focus on today. I'd like to focus on another gradient that occurred where there is this white line right in the middle of the day. You can see that on the left of this white line, there are way fewer deep scattering layers near in the top, let's say 300 meters compared to the other side. So on one side, we have more deep scattering layers than on the other. And also if we look at where certain deep scattering layers are on the two sides, there is almost a shift as we move from one side of the line to the other. So this echogram was measured as the ship was transiting from the eddy core into the eddy periphery. So this gradient was located at the interface between the eddy core and the periphery, and then eventually the ambient waters. And there was this very strong gradient that matched, sorry, I need to move this thing, the transit away from the center of the eddy into the periphery. The black line here is showing the distance from the eddy center, but also match the gradients that we could observe in the near surface. Here there is an example of near surface fluorescence that basically changed dramatically as we moved from the eddy core into the eddy periphery and then outside. Also, the distribution of these layers changed in response of possibly a change in the light environment underwater. We collected some CTD casts with a PAR sensor, so we could estimate light attenuation coefficients, and we also have PAR measurements near the surface. So we could outline an estimate of where the isolums were at different depths. And we can see that when we have deep scattering layers that sort of shifted across the gradient, the light levels at depths changed as well because of differences in the amount of particles in the near surface. So this example showed us that there were strong gradients when moving from the eddy core into the periphery and eventually outside, and these were matching the light properties of the water column and gradients in the near surface. But this is just an example. When we combined all the observations collected through the entire section of this field trip, we found that these patterns were consistent. Here in this figure, we have the daytime average of acoustic backscatter averaged over the eddy core, and this is the red line, and the ambient waters of the subantarctic zone, so the waters around the eddy, and this is the blue line. And they're quite different. In general, there's a lot more sound that is backscattered, especially in the upper mesopelagic between 200 and 600 meters in the ambient waters compared to the eddy core. But interestingly, if we plot the same average for the waters in the polar front zone, so on the other side of the eddy, of the front, where the eddy had originated, we get that is something that is quite similar to what we find in the eddy core. So this is the black line, especially in the upper mesopelagic. So in this region between 200 and 600 meters, we have that the ambient waters have way more acoustic backscatter compared to the eddy core and the origin waters of the polar front zone. And we can see this through differences in profiles, but also if we integrate across the water column, we find a pattern where basically inside the eddy core and in the polar front zone, there is about half of the acoustic backscatter that we find in the subantarctic zone. And this is interesting because it's also consistent with previous studies that found a difference in acoustic backscatter between the subantarctic zone and the polar front zone. So here I'm just showing on the left-hand side some observations from the IMOS database, where we put together all the observations from ships of opportunity and research vessels collected during the autumn months at 18 kilohertz. And we can see that north of the subantarctic front, that is the black line here, generally acoustic backscatter is way stronger than south of that. Our observations confirm that pattern, but also extend that to eddies that have originated on one side of the front and then traveled into the subantarctic zone. So to sum up, this study that where we sampled one eddy, ideally we would do this for many others, showed that the eddy we sampled presented a unique habitat, and it was very different from the ambient waters. The distribution of acoustic backscatter followed really closely the distribution of near-surface tracers, all the way into the mesopelagic. And it looks like the eddy was acoustically more similar to the waters where it had originated compared to the waters that surrounded it. So the question is, why do we get that? Is it possible that there is some trapping? We're talking about micronectons, so these are animals that can swim. What does this mean? So mesoscale eddies have a bit of a reputation for being highly nonlinear, which means that they rotate faster than how they move, how they translate. And this means that if you have a tracer, such as nutrients or heat or salt, that tracer will be transported by the eddy. And here on the bottom, there's a figure showing the nonlinearity parameter, that is a comparison between the rotational speed and translational one, calculated for the eddy at different depths. And generally, the threshold for nonlinearity is 1, that is this black line. And you can see that the eddy we sampled was highly nonlinear, meaning that the trapping power was very high. However, as I mentioned, the animals we're thinking about are not plankton, they can swim. The question is, how much can they swim and why would they swim? So there is a study that came out quite recently where the authors used the acoustics of single targets to look at the swimming behavior of some micronecton. And they found that generally they can move between half to 1 body length per second. And so I sat down and did the math to see if a fish could potentially move from the eddy core all the way into the ambient waters within a month. And technically it's possible, but only if the fish was going straight for the entire time and never stopped swimming at maximum speed, which seems a bit of a big effort, but not impossible. But for me, the key question here is it convenient for the fish to move? Why would they engage in such an energetically demanding activity, considering that the physical properties of the eddy, especially in the mesopelagic, stay pretty similar to those of the origin waters? So I think the key to trying to find out if these animals have been trapped or if there is another mechanism that created this pattern we observe here is something along the lines of trying to understand the swimming and possibly the sensory ecology of these animals. So that's something I'd really love to learn more in the near future. And with this, I think I'm approaching the end of my talk. So thank you for your attention. I'm really looking forward to questions and any discussion you may want to have. Thanks.
Thank you so much. Oh, that was pretty cool. It's nice to see results from the eddy voyage. I always hear Phil talking about it, but then it's good to see something out of it. That's great. Well, I mean, if people have questions, how about you put it in the chat or raise your hand? That's fine too. While we wait for people to think about some questions, I have some. I have a few. Did you sample the eddy too in terms of micronectin? Did you have some net deployments, or was there more, continuous like water sampling and, of course, acoustics?
Yeah, I wish we did that. Unfortunately, the entire micronectin study was something almost as an afterthought. There was a lot of biogeochemical sampling. I think also in the mesopelagic in terms of biogeochemistry. So there are some water samples, but unfortunately no micronectin. And that's always a bit of a challenge because fishing, especially at those depths, is very time consuming.
Yes.
So
you
would need a dedicated voyage for that. But I think that's something that needs to be done.
That would be totally interesting also in terms of what zooplankton is there, because I mean maybe they will be trapped because there's a lot of zooplankton in there, and I mean by moving if there's a lot of food around, right? So it would be interesting. I mean, not sure if it's logistically doable, because as you said, it is really, you want to sample the eddy and it's time consuming to put out the nets, but it's definitely something that it's already good to have the data of the acoustics. So it is, even if it's an afterthought, it's definitely a good use of the data we collect on every voyage. So, and the other thing is, it's really interesting that there's low backscatter in the polar frontal zone and an Arctic zone compared to the subantarctic, because it's the same thing we observed on the Solos voyage in 2020. We had the same thing with lots of stuff going on in the subantarctic. It was during summer. But there was lots of stuff going on in the subantarctic with lots of fish migrating, and then once we went south of the polar front, there was nothing going on. We didn't sample particularly eddies, but still it's interesting pattern to see. I guess it's just a different community or something that is driving things, but yeah, it's definitely interesting.
Yes, one of the questions for me is because acoustics can be used as a sort of proxy for biomass. In truth, you're only seeing the reflecting sun.
Exactly.
So there's a paper led by Tracy Dornan where she actually collected samples and looked at the swim bladders of the fish on the two sides, not on the two sides of the front, but going from probably the subantarctic zone all the way to Antarctica. She found that while acoustics was going down as you approach Antarctica, the amount of fishes that you caught was kind of the same, but they had a different physiology with the swim bladder. So I think that the trapping story would still be there, but it would be more a matter of community composition rather than abundance. But I think then for me, the other big question is, we're focusing a lot on fish, but I suspect there's a lot of squid here, and that's really hard to catch with nets. So I think I'd love to talk to squid people and see if they have any suggestions, because it would be really something.
100%. Yeah, 100%. Oh, yes, super interesting. In our case, in the polar frontal zone, we had lots of salps too, so they wouldn't really appear on the, well, they appear, but not as consistent as fish, and there were still lots of biomass in the water, but not visible really. So yeah, it's another story. It's super interesting. Did anybody else have a question? How about the audience?
Catherine has a raised hand.
Katie, sorry, I didn't see that. Katie, go for it, Katie.
Okay, can you hear me? Hi.
Yes.
Okay, thank you so much, Ali. That was really interesting. I had a question about your backscatter figure with the NASC. There.
This one?
I was just wondering what, because there's a big difference there's a spike in backscatter between 0 and 200 meters in the eddy core and the origin waters, and I wondered if you had any ideas of what that is, or why there's that difference.
So I don't have a definite answer, but I have a few thoughts. One of them is when I think about the coherence of eddies. Eddies have different ways to exchange waters with the outside. One is at the eddy periphery, sort of through the sides, and that's all connected with the fact that the eddy core is non-linear, but the periphery is way more complex, so you can have those filaments and exchanges. But there are two other ways an eddy can exchange material with the outside. One is in the very deep, where the currents become weaker and then the nonlinearity sort of breaks. And the other one, I think, is near the surface, where you can have that the wind moves a sort of slab of water on top. Mm-hmm. So I suspect that if you look at that peak around 100 meters, it's sort of halfway between the ambient waters and the origin one. So I suspect that's a sort of area where there have been some exchanges with the outside. So that's one option. The other option is that maybe the animals weren't exchanged, but because that area is closer to the surface, that will have experienced stronger differences in, I think, temperature or mixing, because it had been on the other side of the subantarctic front for at least a month. So I would say I would suspect that happens because it's an area of exchanges. Then whether it's an exchange of the actual animals or it's a change in their prey distribution or just the abiotic environment, that's hard to know. I think something that I'd like to do at some point is have a very simple toy model where you have an eddy and you implement some simple rules and just look at how things evolve in time. Something that would be really cool would be to have a sail drone and put it with acoustics and put it in an eddy and let it stay with the eddy for 3 months and see how things changes. Here we almost have only 2 data points in a time series.
Cool.
Does that answer the question? I rambled a little bit.
No, it's just an interesting feature. So interesting. The sail drone idea would be great.
Yes, let's get all the toys in the water, you know.
Yes, please.
Great, thank you.
Yang, you're next.
Yeah, great talk, Ali. So I was wondering, looking at your fluorescence data, so does that correct for the non-photochemical quenching? Because during the day, the fluorescence can be like decreased because the sunlight and the heat. I mean, usually what people do is they use the fluorescence value from the night, but here you're really trying to compare day and night. Have you correct for that?
So that's a very good point. I'll go to it. I guess there is probably photochemical quenching going on there, but I think it would be happening both before and after this transition. So I wouldn't trust the absolute values of the fluorescence. Actually, Seb Moreau, who was on the trip as well, had a bit of a crack at seeing if he could turn this fluorescence data into chlorophyll or something more elaborate. But then we decided to be really cautious and just use that as a tracer, assuming that the quenching wouldn't change between the 2 sides of the white line, which is true up to a point, because of course the day goes by, so you have different intensities of light hitting the water. But I think over that little difference in time, it shouldn't be affected by that. We had a look at the PAR measurements as well, just to check that it wasn't super sunny before and super cloudy after, and it wasn't, so we were quite reassured by that. But that is a good point.
But the jump is really obvious, I guess. I'm just like curious. So you don't have chlorophyll or POC measurements, but you're looking at fluorescence. But it's really obvious. I'm just like wondering. Thank you.
Yeah, no worries. Thank you.
Excellent. And I think let's move on to Corinna. But thanks, Ali, for this amazing talk.